Hi class, my name is Sarah Wilkins. This is the Advanced Group Concepts presentation number 8. We're going to talk today about nucleotide synthesis and a few other things. So for this assignment, we're using the poster that we've made over the past 4 weeks, and I'm going to kind of walk you through the process for how nucleotides and ribose are made and kind of show you how those come together. And those come together to make ATP, and this ATP is then going to be used for DNA replication. So we're kind of putting together everything that we've learned in the past 8 weeks, and I'm going to walk you through that ATP regeneration process. Okay, so today we're going to analyze the pathway and kind of describe that a little bit more, and as well as that secondary reaction that occurs, and whether or not a reaction uses ATP and makes a source out of that, okay? First, I wanted to show you guys a collaboration of what we've worked on within the past 8 weeks. So as you can see, been very busy, and this should look similar to what everyone else has done in the class as well. So we've done pretty much a few sheets each week, putting together different structures and processes and a bunch of different drawings and kind of put them together to make one big process presentation. Okay? Alright, so first of all, we're going to talk a little bit about nucleotides. Nucleotides can be synthesized by a variety of means. This is going to be both in vitro and in vivo. So as far as the in vitro, this is going to be referred to as protecting groups that may be used during laboratory production of nucleotides. A purified nucleoside is protected to create a phosphoramidite, which can then be used to obtain analogs, and these analogs are not found in nature, and or to synthesize an oligonucleotide. Alright, and then I wanted to kind of show you guys this structure. This is something that's going to play a role in these processes that we are here talking about today. So in vivo, nucleotides are going to be synthesized de novo, or recycled through different pathways, okay? So the components that are used in de novo nucleotide synthesis, those are derived from biosynthetic precursors of a carbohydrate and of an amino acid metabolism, as well as from ammonia and carbon dioxide. The liver is the major organ of the de novo synthesis of all 4 nucleotides. So we also want to keep that in mind. The de novo synthesis of pyrimidines and purines follows 2 different pathways. So for the first one, the pyrimidines are synthesized first from aspartate and from the carbamoyl phosphate in the cytoplasm, and that is to the common precursor ring structure orotic acid, which a phosphorylated ribosol unit is then covalently linked. This is explained a little bit better here in this chart, so you can kind of follow along and see as we go through with that. We also want to keep in mind that purines, however, are first synthesized from the sugar template, onto which the ring occurs. So the synthesis of the purine and the pyrimidine nucleotides are carried out by several enzymes in the cytoplasm. A little bit about synthesis of ribose as well. Ribose is a type of sugar that is normally made in the body from glucose. Ribose plays an important role in the synthesis of DNA, RNA, and the energy-containing substance adenosine triphosphate. Okay, so ATP. Another structure that should be familiar to you as well. So a little bit about the pentose pathway. This is also very important. It's called the phosphogluconate pathway and the hexose monophosphate shunt. Okay, so this is a metabolic pathway that is similar to glycolysis, which we're very familiar with. It's going to generate NADPH and pentoses. And most of the ATP produced by the aerobic cellular respiration is made by oxidative phosphorylation. So this is going to work by the energy released in the consumption of pyruvate, used to create potential.